Great. Well, good afternoon, everyone. I really appreciate the opportunity to participate today and thank the prior speakers for really terrific presentations. So what I'm going to share with you today is a collaboration with this team and my lab and Danny Duick's group at the VRC to discover antibodies against West Nile virus. All right, here. So the work at the VRC is led by the following group. So Danny is my partner in this project. Within my laboratory, the virology work is led by Kim Dowd, the antibody discovery work led by Catherine Burgomaster, and the animal studies by Laura Van Varken. The collaboration is managed by Allison Spalding, and we've worked very closely with Amy Henry, who leads the VRC antibody discovery group. So to take a step back, it's important to appreciate that immunity to flaviviruses and West Nile in particular really is a team sport. The innate and intrinsic immunity is probably the most important component in that animals with deficiencies in innate immunity are profoundly susceptible to infection. T cells are important in clearance of infection, and antibodies are a critical component of immunity. And my laboratory focuses on antibodies because antibodies are sufficient for protection when administered prior to infection. So they are capable of preventing acquisition and disease. And antibodies have therapeutic potential, as I will show you in just a few minutes. So antibodies contribute to protection from flaviviruses in multiple ways. The first is antibodies have the ability to inactivate the infectivity of virions through simply binding the virus particle in a process that I'll refer to as virion neutralization throughout my talk. In addition, antibodies contribute to protection via their capacity to orchestrate immune functions and what the field refers to as antibody effector functions. And this works through interactions between the heavy chain, the constant heavy chain region of the antibody and FC receptors on immune cells such as macrophages. So it's important to note that not all protection actually requires neutralization to occur. The third thing, which I'm not going to spend a lot of time talking about today because it has no direct relevance to West Nile, but is something that is a theoretical thing to always keep in mind when thinking about flaviviruses, and that is the demonstration that antibodies appear to have a mechanistic role in increasing the severity of secondary dengue virus infections. This occurs only in a small fraction of secondary dengue infections, but this mechanism called antibody-dependent enhancement infection is an antibody concentration increase in the efficiency of virus infection that requires interactions between the antibody and FC receptors. And so thinking about antibodies and antibody therapeutics and potential cross-reactive antibodies, this is certainly something to have in mind as we think about antibodies for passive and therapeutic applications. So flaviviruses are, I'm going to tell you just a teeny bit about background of the virology of these viruses so we can sort of hone in on how antibodies are working through binding the virion. So flaviviruses are positive stranded RNA viruses. They encode one long protein that is cleaved by host and viral proteases shortly after synthesis into three structural proteins, and those are the ones I'm going to talk about today. The two that are on the outer surface of the virion is called PRM and E. This is the E protein here on the right. The E protein I'm showing you as in an anti-parallel dimer, so this is sort of two proteins touching each other in a head-to-tail fashion. This is one monomer, this is the other. The E protein is organized into three different domains 1, 2, and 3. And the key feature of the organization of the flavivirus compared to something like SARS-CoV-2 or influenza is that these envelope proteins lie flat against the membrane of the virion and decorate the virion in this, I think, really elegant herringbone configuration that places 90 anti-parallel dimers on the surface. From the perspective of antibody recognition, if you stare at this virus particle, you'll see that the E proteins on the virion exist in 3 different chemical environments defined by their proximity to this 5-fold symmetry axis, this 3-fold symmetry axis, and the 2-fold symmetry axis. And why I'm sharing this, this detail with you is that depending on the epitope recognized by an antibody, it may be able to bind the virion different numbers of times, depending on the accessibility of that epitope on different parts of the virus particle. So not all antibodies bind the same number of epitopes on a flavivirus. So I'm really excited to have the opportunity to collaborate with this group because the VRC Sheba collaboration has really the potential to contribute greatly in multiple areas, and that includes fundamental advances in understanding flavivirus immunity, discovery antibodies that we could use to create more specific diagnostic tests for flaviviruses, which is a real need. And then, as we will discuss in the coming slides, development of a monoclonal therapy to limit West Nile disease. So I just really want to emphasize how exciting it is to have had the chance to collaborate with Gili and Yaniv and Yael on this project of flavivirus antibody discovery. I think it's really going to be the first of many interesting things we do together in the years to come. So let me tell you a bit about the antibody discovery pipeline that Yaniv introduced briefly. And this is a pipeline that is organized around the common principle that's been known for quite some time, that you can take B cells out of a convalescent human and sort those B cells based on the specificity of the antibodies that are encoded by those B cells. The really innovative part about what we're doing at the VRC is to use a technique that is called RATPIG that Yaniv introduced. And what RATPIG is a very clever strategy of molecular biology that allows you to clone the genes encoding antigen-specific antibodies and assemble them in a test tube in a way that allows you to deliver them into cells and make antibodies that allows you to do functional tests very quickly. An important piece of this is you also get the sequence of those antibodies. So instead of a process that takes the better part of a month, we can sort B cells, do this RATPIG technique, and if you have high throughput functional tests, which we do, you can do antibody discovery very quickly, and that's what we were able to achieve in this project. The functional test we use, I want to introduce because I don't think I'm going to show you any data with a real infectious virus neutralization assay, although we do those, is a technique that I developed while I was a postdoc that is called a reporter virus particle, or RVP. And an RVP is essentially a viral pseudotyping system similar to other technologies used for other viruses. The backbone or the core of this technology is a modified form of the genome in which the structural genes that comprise the virion particle are removed and they're replaced with a reporter gene. So if you deliver this RNA directly into cells, it will express a reporter gene either from the 5' end or the 3' end, depending on how the replicon was configured. But the interesting and useful feature is that you can complement this replicon with the structural genes of the flavivirus and trans. You just put them in the same cell together. They will produce a reporter virus particle that is capable of a single round of infectious entry. And you can measure that infectious entry either using flow cytometry, like I will show you today, or using luminometry if the replicon encodes luciferase. And that is the technology that we transferred to Sheba. So RVPs for studies, you can do a lot with them, and the field now does a lot with them. But for what we'll talk about today, we're using RVPs as a surrogate for an infectious virus. And we're doing this, number one, because it reduces the containment considerations. But number two, the fact that you can read out infection using a reporter gene allows us to do things at very large scale and do so very quickly. But the most important thing to appreciate is that from the perspective of an antibody, an RVP is indistinguishable from a real infectious virion. This is data from Zika virus RVPs that was performed during the Zika epidemic. And what I'm showing here is the results of a neutralization assay with an authentic virus and an RVP when the assays are performed using the same cellular substrate and using the same method to read out infection, in this case, flow cytometry. So RVPs, I think, are a very faithful proxy for virus infection. The other interesting thing is that we can take RVPs and we can make RVPs for a very large number of different flaviviruses, like I'm showing here, or actually a very large number of different strains within a particular type of flavivirus, like I will show you in the coming slides. And just to point out, we've used this technology to read out the immunogenicity data of 5 different clinical trials of Zika investigational vaccine candidates over the years. So let's get to the work that we've done together. This is the screen that Yaniv mentioned, where we screened serum from the Sheba healthcare cohort of samples that were negative by the diagnostic ELISA done at Sheba and positive. And so you can see this is the different degrees of positivity that we scored throughout the sample set. And as Yaniv mentioned, we will ultimately discover antibodies against those with the highest titers. But to just really emphasize how many things could be done together, I wanted to show you this slide because this really is a good cross-section of the type of samples that, the type of patterns of neutralization you can see in samples from individuals that are living in or traveling through regions with multiple circulating flaviviruses. So you can see that some samples had minimal activity. This was a negative sample. This example in the middle is what the field would call type-specific neutralization. It neutralizes West Nile and red, but does not neutralize Zika or Usutu or yellow fever or dengue. But then this sample is from an individual where their serum is capable of potently neutralizing West Nile, but with significant cross-reactive neutralization to Usutu, yellow fever, and dengue. And we could discover antibodies for all of these viruses out of samples like this. Today, we're going to focus exquisitely on the West Nile, but I wanted to show you this because this really emphasizes the the really rich opportunity afforded by collecting such an interesting collection of samples as you've done through the healthcare cohort. So this is on this slide. What we've done here is we've sorted the B cells, and this sorting was done by Danny's team. And then the rat pig procedure was done again by Danny's team. And then we took the samples and we screened them. And I'm showing you really a multi-parameter screen of the supernames of these rat pig gene product transfections. And what we've measured here is virus neutralization on the y-axis using RVPs. Virus binding, this is the y-axis, excuse me, virus neutralization on the y-axis. virus binding on the x-axis. I'm also showing you the degree to which the antibodies in that supername bind a monomeric form of the West Nile E protein, and that's in the colors. And the ability of those rat pig supernames to bind just the domain 3 portion of the E protein. And so what you can see is that we discovered antibodies that neutralize potently and also bind. We discovered antibodies that bind but don't neutralize. We've discovered antibodies that are potently neutralizing that do not bind domain 3, and some antibodies that are neutralizing and bind domain 3. My interest in domain 3 is that the benchmark antibody that we use in our thinking for this project is an antibody called E16. E16 is an antibody discovered in mice that was humanized by my colleague and friend Mike Diamond many years ago. And we have studied E16 extensively over the years, and this antibody binds domain 3. This is this slide is a little complex, but I wanted to include this to illustrate an important thing about thinking about flavivirus neutralization, and that is these black curves detail neutralization activity and correspond to this left y-axis. This red curve corresponds to the ability of these same neutralizing antibodies to support antibody-dependent enhancement in vitro. Now, every virus that you study can do this trick. In vitro antibody-dependent enhancement of infection has no correlation or association whatsoever with what occurs in the field. This is simply a test of whether antibodies can bind the virion, and by virtue of binding the virion and making these aminocomplexes, can interact with Fc receptors on target cells. And we showed many years ago that the number of neutralizing antibodies and the amount of enhancing antibodies is related simply by the number of virions attached to, the number of antibodies attached to the virion. So this is the expected sort of pattern that you would see on a cell expressing an Fc receptor. But I wanted to show you this profile here. This is an antibody that clearly neutralizes infection, but what it does not do is neutralize infection of all of the virions in the tube. So this is important because there are antibodies that neutralize, but they can't bind enough times to neutralize all of the viruses in the assay. And so for that reason, we can measure antibodies that have neutralizing activity, but in our very stringent quantitative parameters for measuring how much neutralization occurs, they score as negative, as you can see here. And that's because they don't neutralize 50% of the virions in the tube. And there are an awful lot of antibodies like this that are produced in vivo. Today, we're going to focus on antibodies like this that neutralized all of the viruses in the tube and did so at low concentrations of antibodies. And I'm really going to focus my comments on just 5 of these antibodies. These were the first 5 very potent antibodies we've studied. We're working up another 5 antibodies of a similar potency now. So this is the EC50, or the concentration of antibody that inhibits 50% of infection for these 5 antibodies that we've just numbered and color-coded. So these antibodies are quite potent. They neutralize at about 10 nanograms per mil or less, which is about best in class for neutralizing antibodies for flaviviruses. And this on the right, what I'm showing you now, we made RBPs out of a library of West Nile sequences that represent the diversity of this virus in nature. On the left are the viruses that are clustered into lineage 1. On the right are the viruses that are clustered into lineage 2. This gray arrow depicts the virus that was circulating during this past year. And what I'm showing you here is the ability of these potently neutralizing antibodies to neutralize this diverse population of West Nile sequences. And you can see they do so to varying degrees. I think the notable antibody 198 among this group is the least potent when neutralizing these lineage 2 isolates. These black boxes are just simply experiments that have not been completed yet. So we have potent neutralizing antibodies. We were interested to understand whether these antibodies had the ability to cross-neutralize other viruses in the Japanese encephalitis virus serocomplex. So this is the serogroup in which West Nile resides. And the answer, to my surprise, is yes. It's that 3 of these antibodies actually quite potently neutralize Japanese encephalitis virus. And you can see this in this raw data, where black is the West Nile neutralization assay and blue is the JEV neutralization assay. They neutralize Murray Valley to some degree, and even St. Louis encephalitis virus. So this is really interesting. This is an example of one of those basic science surprises that, in addition to having potential translational value, is something we're quite interested in understanding further. In this graph, I'm showing you a larger number of antibodies, but just to highlight the point that we're using an instrument called a Cartera, supplemented by 2 other methods to start binning the somewhat large number of antibodies we've discovered into competition groups. This work, all of the data I'm showing you is unpublished, and all of the data I'm showing you is pretty hot off the press, if you will. But what we know so far is that 3 of these antibodies, and these are the ones that had the ability to neutralize JEV, are in a single competition group that is distinct from this 198 antibody that does not compete, but is in this part of the competition group. And this antibody 215 we have not yet mapped. So we have at least 2 different competition groups so far among the potent antibodies we've studied. Another way to map antibodies is to try and select for neutralization escape variants. You do this by infecting viruses in the presence of relatively high concentrations of antibodies, but not enough that you block all infection. And this provides selective pressure for the virus after 3 to 5 passages to select something that allows it to escape from neutralization. So we did this for all of the antibodies. These are the 3 antibodies for which these are the data for which we have the highest resolution so far. What I'm showing you here is not the neutralization escape experiments, but reporter virus particles that encode mutations identified by neutralization escape experiments. So this S276N mutation, which is at this junction between domain 1 and domain 2, confers escape for this antibody 198, but has no impact on antibodies 209, 211, or 213. In contrast, the virus adaptation at position 67 or 68 confers either complete or partial escape from neutralizing antibodies 209, 211, or 213. And these are all the antibodies in that same competition group, which really makes some sense. So we now are starting to have a general idea that antibody 198 binds in this region somewhere, and these cross-neutralizing antibodies binds at this distal end of domain 2. And as I mentioned, these patterns of escape are shared by these antibodies that have a shared competition group. Another way to think about how antibodies bind is to start to ask, what is the requirements for antibody binding with respect to the oligomeric state of the E protein? So I mentioned this is the E protein, and on the virion, it's in this anti-parallel dimer format. And it's known that most antibodies actually have the ability to bind epitopes that are on a single E protein. Some antibodies that have been discovered, and increasing numbers of them are now being discovered because we're looking for them, bind epitopes that are composed of more than one surface. So perhaps across this dimer interface. So it takes more than one protomer for antibody recognition. This is E16. E16 can bind dimers of West Nile 1 and 2, but also quite potently binds the monomer. Antibodies 209, 211, and 213, these antibodies bind the monomer pretty well. In this study, you can see 213 efficiently binding the JEV monomer. We're kind of curious why we didn't see that with these other two antibodies. We're still looking into that. But 198 and 215 clearly don't require a monomer for recognition. And so that suggests that perhaps this epitope might be a quaternary surface, maybe the antibody binding across the dimer, which has been seen for other viruses. The last bit of thinking about how antibody recognition I want to share is that, as it turns out, flaviviruses don't all look exactly the same. This is a EM image taken by Richard Kuhn and Michael Rossman, my colleagues and friends at Purdue. And what you can see is this is what a mature virus looks like. But there are these other viruses in the field, like this one that has a spiky surface on one end that corresponds to the immature virus, an example of what you can see here, but then a smooth surface on the other end. And over the years, my laboratory has very clearly demonstrated that these different structural forms of mature and partially mature viruses are not only infectious, but interact with antibodies differentially, such that in most cases, the process of maturation reduces the potency of neutralization, and you can see that by the shift to the right of the gray curve relative to the green curve. Or in one example, thus far, an antibody we discovered with Jim Crow many years ago, we discovered an antibody that more efficiently recognized the mature form of the virion and less efficiently recognized the partially mature form. The take-home message is that the epitopes on this smooth surface are harder for antibodies to get at, whereas antibody accessibility is greater on the spiky surfaces. This is data from experiments we did many years ago with Zika that formally demonstrates that for Zika, it's the antibodies that bind the mature surface of the virion that correlate best with protection. And that is demonstrated in an experiment where we immunized mice with 2 vaccines. We normalized we collected serum, pooled it, and normalized its activity to the ability to inhibit the standard population of viruses that emerge from cells, or a mature population of viruses that we can produce in the laboratory by making RVPs in the presence of furin, passively transferring that serum into mice and then challenging them. And when you normalize infection by the level of mature neutralization, you can see that 2 vaccines perform similarly if you normalize them by standard preparations, standard neutralizing activity. You can see these 2 vaccines did not perform similarly, and this difference in protection here is not observed when you measure neutralization by standard assays. So these 2 vaccines elicit the same exact level of standard neutralizing activity. So this was an experiment we used to drive home the point that neutralization assays don't always tell the truth. And in this case, we had to determine that it was a neutralization assay focused on the mature form of the virion that correlated with protection. So I was quite interested in understanding whether antibodies, as we discovered them, I think the ability to neutralize mature forms of the virion is a key property we're looking for. And you can see that by comparing the circles to the triangles. So this 198 antibody sees them both equivalently. Some of the antibodies we can see more efficient neutralization against mature forms. So these are all good cues that the 5 antibodies that we've discovered have this positive characteristic of what you might think of as a target product profile for a monoclonal antibody for use in therapeutic or passive protection. So last few bits of data is the animal studies. So in this experiment, after standing up a lethal West Nile virus model in mice at the BRC and C57 Black 6 mice with 100 PFU of the New York 99 strain of West Nile, we were able to demonstrate lethal infection in the presence of a control antibody. This is an antibody we recently discovered against the lacrosse virus. And then every one of the antibodies that I have shown you today, including E16, will protect all mice from infection when administered 1 day prior to infection. This is, in my mind, expected from the in vitro data we have. As you can see here, in this model, we're getting viremia at day 2, and we're preventing viremia, the acquisition of infection, through the use of these antibodies. The introductory comments, I mentioned that antibody Fc effector function can be important. To begin to study this, we modified each of these antibodies to introduce 2 mutations that are referred to as the LALA mutations. These mutations greatly reduce but don't fully eliminate the ability of antibodies to interact with Fc receptors. And you can see that the level of protection is identical. I will share that the LALA variants not only block antibody effector functions, but have been shown in pretty sensitive models of secondary dengue infections, or a dengue ADE model, that the introduction of a LALA mutation abrogates the ability of antibodies to support antibody-dependent enhancement of infection. So there is a reason why a potential West Nile antibody product might include a modification of this type. So this is protection 1 day when the antibody is delivered 1 day before infection. On this slide, I'm showing you our ongoing studies to deliver the antibody 1 day after infection, 3 days after infection, or 5 days after infection, to start looking at the sort of intent to treat capacity of an antibody in a model like this. These experiments are not all done with the same dose of antibody. This is 50 micrograms of antibody, 200 micrograms of antibody, 500 micrograms of antibody, and 2,000 micrograms of antibody, and consistent with how these experiments are done by others in the field. It takes more antibody to get the antibody into the brain to protect the animal when you deliver the antibody so close to when you start to see lethality in this model. And so what you can see is that we have really almost complete protection with except 1 mouse in all of these experiments, either the wild type antibody or the LALA, all the way through application 3 days prior to infection. And now at day 5 after infection, we're starting to see breakthrough infections in this experiment. These experiments are still ongoing. Many of these experiments are currently being repeated to increase the number of mice in each experiment, but certainly the data demonstrates that these antibodies that we have discovered together can protect infection. And so these are the characteristics that I've sort of walked through that I think are really important in thinking about an antibody as a therapeutic. We have discovered antibodies with high in vitro neutralization potency. They can neutralize diverse contemporary and regionally important West Nile viruses. They recognize structurally mature forms of the virion, and they inhibit infection when administered post-challenge. So what's next? What are we doing? We are doing additional studies of potent antibodies. We're continuing to look for better antibodies. Of course, we could do that for a long time, but if anything better in this first group of antibodies we've studied. We are doing additional animal studies to repeat things I've shown you or do dose de-escalation studies of the best antibodies in the group to see if we administer lower amounts of antibodies, can we start to differentiate all that protection I showed you on the prior slide. And we're looking at PK for these antibodies, although PK may not be important depending on the application. We're studying antibody affinity and starting to do structural studies, and we're continuing to do resistance mapping to really understand the sensitivity of these antibodies to variation once we know where their epitopes are. So in my final comments and thinking about down selection, this is a little bit of a complex, set of data to put together. And that's because we know many of our experiments are done with this New York 99 prototype strain, which is a lineage 1 virus. They're very potent. When we look at all of the strains of West Nile in our studies, you can see they're all quite potent, but there are viruses that the antibodies are incapable of neutralizing, and this information is potentially important. And then we're starting to see at this very stringent model of 5 days post infection differences in the level of protection. These are experiments, of course, that are done with New York 99. And here showing you that we have things to think about quaternary recognition, the cross-reactive, cross-neutralizing responses that also sort of play into how we will think about and discuss together, once we have all the data, what is the most interesting antibody. I would say that, if I had to choose today, I think there's really two options here. I probably would set aside 198 because of this significant breadth of its, this significant differences in its ability to neutralize broadly West Nile viruses, including those lineage 2 viruses. And so it's really down to, I would say, this one competition group and 215. 215 is an attractive antibody. There is a virus or two that is in lineage 1 that it misses entirely, and we're really interested in understanding at a molecular level exactly what amino acid change confers this escape phenotype. That will be an important piece to deciding whether 215 is a promising candidate. So that's it for me. I want to really first start thanking thanking you and my colleagues Gilly, Yaniv, and Yael, who are providing this really terrific opportunity to work together on a really important problem. I had the privilege of meeting Yaniv many years ago. I had no idea that this collaboration would materialize as I transitioned to the VRC, but it was a great benefit of my new opportunity. My laboratory now is called the Arbovirus Immunity Section. Virtually my entire laboratory is working on this problem. It is on the front of our minds. I have the great privilege of working with Danny and his laboratory on this project. Allison Spalding, who is a leader of our premise program, and Amy Henry's team, who does the antibody discovery work, and Leo Cibriani at our vaccine immunology program, who has contributed in multiple ways to the work. And finally, I want to thank Steve Whitehead. Steve is notable as the inventor of the NIAID tetravalent dengue vaccine and the scientist behind the 3 chimeric West Nile vaccines that was in Yaniv's slides. And we got the strain of West Nile we used in these animal studies from Steve. So let me stop there and take any questions.
Questions, anybody? Beautiful data. It's really exciting, I have to say. Yes.
Can you elaborate a little bit about the role of, if there is a role of pre-fusion and post-fusion conformation in flaviviruses, and does it can it affect your screening method?
Did you hear?
Yeah, I could hear the question. Yeah, thanks for the question. I think I included the slide to show you how complex serology is for flaviviruses, in part because it is an aspect of the field that I think could be improved. Neutralization assays in general are the most specific way you could diagnose, determine what the infecting virus was that gave you flavivirus reactive antibodies in serum. Of course, the challenge is that these are complex experiments that are not, in most hands, rapid, so they're not available to use in diagnostics at the bedside, if you will. We can see by measuring large numbers of flaviviruses in any given sample, we can see what is the rank order in titer, if you will, for a variety of different viruses. For example, there was one sample that we studied that might have been a Zika infection. The Zika neutralization titers that in the samples we received were higher than the West Nile titers, and that might have been a Zika infection that was either most recent or contributing cross-reactive neutralizing antibodies to the rest of the assay. And then certainly, the more flavivirus infections you have, the more cross-reactive your serum is. So the power of antibody discovery is that instead of trying to deconvolute serology by looking at new curves and imagining what the sequence of events was, we will just clone individual B cells, and we will look at the antibodies that are encoded by them. For this project, one, I think, really great opportunity we have is that we will also start to get the genetics of the West Nile antibody response in humans. We have a large number of samples, and we will continue to sort and do sequencing to really understand the genetic signature of what a West Nile virus antibody response looks like in humans, and then assign each of those genes to the types of antibodies that they encode. I hope that addresses your question.
Thanks. Yeah, next question.
Can you please elaborate regarding the mice experiment on the route of infection and the route of treatment, and if the treatment had an influence on the viral nodes in the brain, for example?
Yeah, thanks for the question. Where all the infections are foot pad. We haven't. That's the traditional model. We did none of the experiments, for example, or IC inoculation. We haven't explored whether if you gave the virus other routes, whether we would get a different result. I think the foot pad is probably the right place to do this. We know Heather Hickman at the NIH has been doing a lot of work with flaviviruses now and sort of mapping precisely what happens and where the virus goes and what are the order of events that occur when you deliver a flavivirus into a mouse in terms of the order of where the virus replicates and the contribution of replication in those compartments to overall viremia. I think we're on the right track, but we certainly can do other things when time permits.
Yeah, please.
Thank you. Amazing data. So I have two questions. One is, do you have any information about allele usage, or from how many patients, different patients, these antibody sequences are coming from? What is the degree of maturation? So something about their CDRs or distribution between quaternary and linear epitopes. And the second question is whether you have any structural data on quaternary epitopes recognition, either priam or histography. Yeah, so let me go backwards. We will obtain structural data on the top few antibodies that we have discovered. Certainly, from what I've shown you, we will look at least one of those antibodies that has that cross-neutralizing activity that is a non-quaternary surface, and then both of the antibodies, 198 and 215, that have
quaternary surfaces. Those are interesting from many levels. In terms of the genetics of the antibody response, I think I want to punt that question until the next time I have the chance to present. We are getting that data. We have all the antibodies that I have shown you today are from one of two people. We have opportunities to do larger numbers of them, and I think we will end up with a very rich data set about the genetics and about the degree of mutation required to achieve different levels of activity. We don't have a ton of that data yet, and it hasn't really been analyzed, I would say, in the way that you're interested in. But I'm interested in that too. So that was a good question. Thank you.